Namaste. Today we will start our next module, which is conservation genetics. In this module we will have 5 lectures. The first one is preliminaries and introduction to genetics, followed by population genetics, chromosomal and genetic disorders and inbreeding, population viability analysis, reintroductions and outbreeding. So let us begin with the first lecture, preliminaries and introduction to genetics. So let us begin with what is conservation genetics. Now we know what conservation is, we have dealt with in the first module. Now conservation biology, the biology of conservation plus genetics gives us conservation genetics. Now we can define conservation genetics as conservation genetics is the branch of science that aims to understand the dynamics of genes in populations, principally to avoid extinction. We need to look at this definition in more detail. It is a branch of science that aims to understand the dynamics of genes in populations. So essentially we are concerned with populations of animals. Now populations is the group of animals that are living together of the same species. Now they will be having genes inside them, we will come to genes in a short while, and these genes function in dynamic ways. So essentially we are talking about population genetics, or how genes are changing from one generation to the next in the same population. And once we understand their dynamics, we can use that knowledge to avoid extinction. So conservation genetics is the branch of science that aims to understand the dynamics of genes in populations, principally to avoid extinction. So our aim is to avoid the extinction of the population of species. So then we come to the definitions of genetics and gene. Genetics is the study of heredity and variation of inherited characteristics. The study of heredity. Now heredity is how different traits move across different generations. So basically when a child is born, then you would often hear statements such as this child has the eye of his mother, or this child has the hairs of its father, or things like that. So essentially the traits that were there in the parental generation also express themselves in the child. So this movement of traits across generations goes by the name of heredity. So genetics is the study of heredity, how this thing is happening, and the variation of inherited characteristics. So basically everybody of us has inherited certain characteristics, but all of us are different. So all of us have different heights, different colors of skin, different colors of eyes, different colors of hair, whether the hairs are straight or whether they are rounded, and so on, whether they are curled or whether they are straight. And when we look at their cross sections, whether they have a flat cross section, a rounded cross section, or an oval cross section. So all of these are different inherited characteristics that vary between different organisms or different individuals in the same population. So genetics is the study of heredity and the variation of inherited characteristics. Gene on the other hand is a unit of heredity which is transferred from a parent to offspring and is held to determine some characteristic of the offspring. So when we talk about all of these different characteristics, the height, so we will say that there could be a gene for a height, because of which this character of height becomes an inherited character. Whether a child has curly hair or whether he or she has straight hair, so we would say that there must be a gene that is determining whether the hair is curly or whether the hair is straight, because of which this is being transferred as an inherited characteristic. So gene is the unit of heredity. Now in strict biological terms we could even say that gene is a distinct sequence of nucleotides forming part of a chromosome, the order of which codes for a molecule that has a function. So essentially when we say that there is a gene for height, so how is this gene impacting the height of the organism? So this gene would be a sequence of nucleotides. So this gene would be having some sequence of nucleotides, and this gene, where is it located? It is located in a chromosome, and the order of these nucleotides or the information that is there in the gene is used by the organism to code for something, especially for a molecule that has some function. So basically this gene could be, for instance, coding for a molecule that impacts height. So in that case we would call it that a gene is a distinct sequence of nucleotides forming part of a chromosome, the order of which codes for a molecule that has a function. Then when we say chromosome, so then what is a chromosome? Now before we go to a chromosome, let us understand how our cells are made. So in the case of all of our cells, there will be an animal cell. So there would be a nucleus inside. So this is a cell membrane, this is a nucleus, and then we would be having a number of other things like cytoplasm, which is the sap that is filling the cell. We might be having a few mitochondria, which are organelles where energy is produced. So this is mitochondria. We might be having an endoplasmic reticulum, so which is a distinct structure that is used by different organelles to move things from one part to another. We could be having Golgi bodies and so on. But for the case of genetics, the only thing that we are interested in right now is the nucleus. So we can forget everything else. So there is a cell and there is a nucleus inside. Now this nucleus will be having a number of fibers inside that remain entangled for most of the part. But when this cell is dividing, so when the cell is moving from one cell to two cells, then all of these fibers condense together to form things that go by the name of chromosomes. So what is chromosome? Chromo means color and soma means body. So it is a body that has some color. So basically if we take a cell that is dividing and we break that cell, we will get to the nucleus, we break the nucleus again, and in that case we will be able to see the chromosomes. Now in certain cells, in certain animal cells, whenever we are undergoing a cell division, then there are stages in which this nuclear membrane, so this membrane dissolves by itself, so that all of these chromosomes come out into the cell body, into the cytoplasm. So if we look at these structures out there in a microscope, we see there are some bodies and they are having some color, because of which we call them as chromosomes. So now technically we would say that chromosome is a thread like structure of nucleic acids and proteins found in the nucleus of most living cells, carrying genetic information in the form of genes. So now coming back to the drawing board, here we saw that all of these threads, they are in an entangled form. So if you take any of this thread outside, it would be an extremely long thread, and when this thread is condensed, it forms a small completely condensed body which we call as chromosome. So this chromosome will be having, so this is a thread like structure of nucleic acid and proteins. Now proteins bind this thread into the tightened structure or the coiled structure. So a thread like structure of nucleic acids and protein found in the nucleus of most living cells. Why is it most living cells? Because certain cells such as our RBCs, the red blood cells, do not have a nucleus, so they are enucleated. But in most of our living cells we will be having a nucleus that will be having chromosomes inside. So it is found in the nucleus of most living cells, and the function is that it carries genetic information in the form of genes. So coming back to the drawing board, so if this is a long thread that becomes coiled as chromosome, if we take any small portion, so say this portion or say this portion, so all of these small portions or some of these small portions may be coding for certain genetic information. So not all parts of the chromosome will be coding for some genetic information, but such portions of the chromosome that are coding for genetic information in the form of genes will also be there. So chromosome is a thread like structure of nucleic acids and proteins found in the nucleus of most living cells, carrying genetic information in the form of genes. Next we have alleles. So in this class we will be discussing a number of definitions that we will be making use of in the next classes. So an allele is each of two or more alternative forms of a gene that arise by mutation and are found at the same place on a chromosome. For example, capital P and small p represent flower color alleles for a pea plant. So what are we saying here is that an allele is each of two or more alternative forms of a gene. So when we are considering, say, the gene for a height of a human being, so let us say height. So this height gene may come in a number of varieties. So there could be a gene that codes for a very tall individual, there could be a gene that codes for a very short individual, there could be a gene that codes for somewhere in between, and also somewhere in between. So all of these are different forms of the gene. And why do they have different forms? Because they have different sequences. So these alternative forms that arise by mutation. So how do we get changes in the sequences? So here we saw that this is a sequence of the gene. So this is the green portion. Now suppose some part of it was replaced by, say, a red dot. So these sequences are made out of A, T, G and C, which is adenosine, thymine, guanine and cytosine. So these are four different nucleotides that code this this whole chromosome. So for instance, in the case of English language, we say that we have 26 different letters, A, B, C, D up till Z. In the case of this genetic information, we have 4 different letters, A, T, G and C. In the case of computers, we have only 2 letters, 0 and 1. So with just 0 and 1 you can form everything. You can make a drawing, you can make words, you can make music and so on. Similarly, in the case of the chromosome, just by using these 4 alphabets we can make anything. Now when we are having these 4 alphabets, then if there is a change at any location in which one alphabet gets replaced by some other alphabet, then it is known as a mutation. So this is a simplified understanding of mutation. We also have different other kinds of mutations that we will come to in one of the next lectures. But each of 2 or more alternative forms of a gene that arise by mutation and are found at the same place on a chromosome. Why the same place on a chromosome? Because, coming back to the drawing board, even when this portion was changed, even when this letter was changed, but the whole of this gene remains at the same place. So when we consider 2 different chromosomes, so let us say that this is one chromosome, this is the second chromosome, and both of these were having a gene at this location and one gene suffered a mutation. So even then we are having these genes that are coding for the height of the individual at the same location. So here it says that an allele is each of 2 or more alternative forms of a gene that arise by mutation and are found at the same place on a chromosome. Now we represent alleles with alphabets or groups of alphabets. So for example, capital P and small p represent flower color alleles for a pea plant. So we would say that the color of the flower in a pea plant could be purple or it could be white. So we would say that purple we will represent as capital P, and when another form of this allele is found, we will represent it by small p. If we had even more number of forms, then we could be using things like P1, P2, P3 and so on. So all of these will be referred to as different alleles of the same gene, the gene that is coding for the flower color of a pea plant. Now next is trait. A trait is a genetically determined characteristic caused due to the presence of some allele. So these are 2 different ways of representing the same thing. So trait is the flower color of a pea plant, and this trait is caused by genes, and these genes come in different varieties which we call as alleles. So when we say that they are found in the same location, we can look at it using McFish. McFish stands for multicolored fluorescence in situ hybridization. So here we are looking at chromosome number 3, and in the case of chromosome number 3, the areas that are having the same or very similar sequences of nucleotides in them are represented by different colors. So all of these chromosomes come in 2 pairs. Why a pair? Because one of these chromosomes will be coming from the father, one of these chromosomes would be coming from the mother. Now, in the case of the father chromosome and the mother chromosome, in both of these we are seeing that these colors, pink, red, yellow, dark yellow, pink, red, yellow, dark yellow, and this white on the top, all of these come in the same sequence. Why? Because this region, suppose this is coding for a height gene, so the height gene in the chromosome number 3 coming from the father will be having will have a position here. So in the case of the height gene coming from the mother in chromosome number 3, it will also be having the same position. We won't be having a height gene here and a height gene here. So these come in pairs. Next we have a genotype. A genotype is the genetic constitution of an individual organism. So what sorts of alleles are present in that organism is represented by the genotype. For example, capital P, capital P. Now in this case, what we are saying when we say capital P is that this individual or got the allele for purple color from the mother and also for the purple color from the father. In case this individual had suppose a capital P from one parent and a small p from another parent, so we would say that this is a purple allele and here we have a white allele. White allele, both of which are coding for the same trait, that is the color of the flower. Now in case we have capital P or small p, we call these individuals as homozygotes. Homozygotes for the gene that is coding for the flower color. Homo means same and this is a homozygote because both of these alleles are the same. But if one of these was capital, one of this was small, we will call it a heterozygote. So genotype is the genetic constitution of an individual organism such as capital P. Now phenotype will be the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. So for instance, this capital P in the presence of a suitable environment will give purple colored flowers. Now why is environment important here? Because suppose there is an individual and the mother is very tall, the father is also very tall, and this individual is homozygous for the tall gene. So essentially this individual is having all the capabilities that it should grow up into a very tall individual. But then suppose this individual did not get proteins during its childhood. So essentially this individual was suffering from a starvation. So during those starvation periods, protein is required for building up the muscles of the body, and basically if this individual lacked protein, so it was not able to grow very well. Or suppose this individual was lacking calcium, or maybe this individual was infested with some parasites which did not allow it to absorb all of those nutrients that were there in the food. So this individual, even though he or she is having the genetic constitution that would code for a tall individual, but in the presence of an environment in which he or she is not getting sufficient nutrients, he or she may turn out to be a dwarf individual. So which is why we say that phenotype is the observable characteristics of the individual resulting from the interaction of its genotype with the environment. Now on the other hand, if this individual was having short mother and a short father, and its genetic constitution was also coding for a short individual, so even if you provide this individual with lots and lots of nutrients, this individual would not grow up to be a tall individual. So which is why we say that in the case of a phenotype, both the genetic constitution and the environment, both are important, and it is an interplay between both of these that tells us what sort of a phenotype would come out. Now from here we move on to the Mendel's laws of genetics. Now Gregor Mendel was a monk, an Austrian monk, who first figured out the different laws of genetics. So the first law is the law of dominance. So recessive alleles will always be masked by dominant alleles. For example, purple capital P flower trait is dominant over the white or the small p flower trait. Now what it says is that if you have an individual that is capital P, so the first allele is coding for purple, the second allele is also coding for purple. So in this case you get purple flowers. If you have a small p, so let us write it like this. So the first one codes for white, the second one also codes for white. So in this case we get white flowers. But what do we have when we have a capital P with a small p? So here this one is coding for purple and this one is coding for white. So in this case, do we get individuals that have a color that is somewhere in between purple and white, or do we have situations in which sometimes it will show purple, sometimes it will show white? So the law of dominance tells us the answer to this question. It says that whenever we have two alleles and one allele is dominant, and we generally represent dominant with a capital letter, so anything that is dominant will mask over the phenotype of the recessive one. So in this case, because capital P is dominant, we will be having purple flowers only. Law of dominance says that recessive alleles will always be masked by the dominant alleles. So if you have capital P and a small p, you will have the expression of the capital P. Next we have the law of segregation. The two alleles of a gene separate or segregate during gamete formation, so that a sperm or an egg carries only one allele of each pair. So what it says is that suppose you have a parent that is capital P and this parent is giving out sperms. So each of these sperms would be having a capital P, but if everything goes well, if there is no abnormality, we will not be having a sperm that has two copies of this capital P or a sperm that does not have any copy of this allele. So this is the law of segregation. And secondly, both of these alleles, so let us represent it as by a red color and by a purple color. So both of these alleles are one and the same, but then we are representing them by different colors just to see how they will segregate out. So when you have the sperms, there would be some sperms that carry this allele that is coming from one chromosome, and there will be some that will be having the second allele that is coming from the second chromosome. So the two alleles of a gene, in this case capital P, separate or segregate during gamete formation. So both of these become separate, and a sperm or an egg carries only one allele of each pair. So it does not carry both the alleles, it does not carry 0 alleles, and this explains the 3:1 F2 ratio. Now what is this F2 ratio? F2 stands for, or F stands for affilial generation. So we will come to this here. So the example case is you have done a cross between two individuals. One was having capital P and the second one was having small p. So what do we mean by this? When you had an individual with capital P, then this individual was having purple flowers, and when you had an individual that had a small p, you were having an individual that was having white flowers. And we are beginning with this example case in which we are assuming that both of these individuals are homozygous, which means that in the case of this capital P, it is homozygous. So both of these are capital P's. It is not a case in which there is one capital and one small. So we are starting with two homozygous individuals. Now when we do a cross between both of these, so let us say that this one is a male plant and this one is a female plant. Now in the case of a pea plant, it is having both the male organs and the female organs together, but for the case of this example, let us say that we have different plants, one is a male plant and one is a female plant. So in the case of the male plant, the sperms will be having capital P and capital P, because two of the sperms will be having this capital P and the two of the sperms will be having this capital P. Now in the case of this female plant, we will have the 4 eggs that are small p and small p. These come from one chromosome and these two come from the other chromosome. Now when there is a cross, so this is how we represent a cross, this is known as a Punnett square. So we represent the gametes from one parent on the left side, we represent the gametes from the second parent on the top. So in this case, one parent gave us capital P and capital P, and the second parent gave us small p and small p. So when both of these gametes come together, So we have an individual which will be getting a capital P from the father and a small p from the mother. So in this case, this individual becomes capital P small p. Similarly, this individual also gets a capital P from the father and a small p from the mother. So it is capital P small p. So similarly, all the other individuals are also capital P small p. So in this case, we will say that in the first generation, so in the first filial generation, that is the result of this cross, all of these individuals are having a genotype of capital P and small p, all of these, and the phenotype will be purple flowered because capital P will dominate over the small p. Now, why do we call it a filial generation? Filial means brotherhood, brotherhood or sisterhood, or individuals that are siblings. So this is the first sibling generation. So all of these are together. Now if we perform a cross between the individuals in the first case, so in the first case we had all the individuals that were capital P and small p. Now we are doing an inbreeding experiment here in which we are breeding between both of these. Now the law of segregation will say that suppose this is the male plant, so the male plant will give out sperms in this fashion. So we have a capital P and a small p. So when it gives out sperms, we'll have 2 that will be having capital P and 2 that will be having small p, which we are representing here by putting it on the left side. So one is having capital P, one is having small p. So there are only 2 different kinds of sperms that are being produced. Similarly, in the case of ova, we also have only 2 different kinds of ova, capital P and small p. So what happens when both of these come together? So this is capital P from father, capital P from mother. This is small p from father, capital P from mother. This one is capital P from father, small p from mother, and this is small p from father and small p from mother. So we have a genotypic ratio of capital P as 1, capital P small p as 2, and small p as 1. So we have a genotypic ratio of 1:2:1. Now if you look at the phenotypic ratio, what will these individuals look like when they grow up? Will be having purple colored flowers. This capital P will give us a purple colored flower, and so will a capital P and small p. So we have 2 plus 1, 3 individuals that are having purple flowered and only 1 individual that is having a white flower. Now in the case of genetics, when we talk about these ratios, these are an averaged out ratios. So it is not necessary that if you are having just 2 individuals, you will be having these 4 different phenotypes. But then if you are doing these crosses in a large numbers and then you take averaged out ratios, you will get this ratio of 3:1. The next law is the law of independent assortment, which says that each pair of alleles segregates into gametes independently of the other pairs, and this explains the 9:3:3:1 F2 ratio. Now what do we mean by this? Each pair of alleles segregates independently of the other. So in this case, we are considering individuals that are having 2 different traits. So we have this flower color, which is given by P and small p, and we have pod color. So in the case of a pea plant, we get pods. So there is a pod inside which we have the seeds. So what is the color of the pod? Now in this case, we have colors as green and yellow. Now green here is dominant, so it is written as capital G, and yellow is recessive, so it is written as small g. Now in this case, when you have this individual capital P, capital P, capital G, when it gives out sperms, what will different kinds of sperms will be formed? So the first is capital P, capital G, the second is capital P, capital G, the third is capital P, capital G, and the fourth one is again capital P, capital G. So we are getting only one kind of sperm that is having capital P and capital G together. Similarly, when we consider the second case in which we have small p, small p, small p, small g, we are getting only one kind of ova, that is small p and small g. Now when we say that both of these alleles are separating independently of each other, that is independent assortment, we mean that when we consider this case of capital P, capital P, capital G, then if we are considering this capital P, then whether this sperm gets this G or whether this sperm gets this G is immaterial. So most of our situations will consider that this P can come with this G, or this P can come with this G, the second G. So this is independent assortment because these alleles are separating independently of each other, they are not coming as a combination. So they are not coming as a combination like this P will always come with this G and this P will always come with this G. There is no such combination, they are coming out independently of each other. So now when we look at the Punnett square, we have only one kind of ova and only one kind of sperm, and so we have only one kind of individual, that is heterozygous for both of these alleles. So capital P small p and capital G small g. The genotype is the same for all the individuals and the phenotype is also the same for all the individuals. So capital P dominates over small p, so we get purple flowers, and capital G dominates over small g, so we get a green pod for all of them. Now in the F2 generation, we are crossing these individuals with each other. So we have capital P small p, capital G small g crossed with the capital P small p, capital G small g. Now in the case of this individual, what all kinds of sperms will we get? Because these alleles are assorting independent of each other, so we can have capital P with capital G, or capital P with small g, or small p with capital G and small p with small g. So we have written all these four combinations here. So these are the four different kinds of sperms that will be formed. So we have capital P, capital G, capital P small g, small p with capital G and small p with small g. Now if these alleles were not assorting independently of each other, so in that situation we would have a case in which in the first generation now suppose these two come together always, so we would be having capital P and capital G, and this would give us small p and small g. Now when we have a cross, we get an individual that is capital P small p, capital G with a small g. But in the next generation, this would not give us four different kinds of sperms, but would only give us two different kinds of sperms, because capital P and capital G always come out together. So it will give us capital P, capital G and small p, small g. And similarly in the case of the mother plant as well. So if that were the situation, we would be getting capital P, capital G, small p, small g, capital P, capital G, small p and small g. So making out the Punnett square, we will have these individuals that are completely homozygous, and these are the two individuals that are heterozygous. And in this case, we would be getting genotypic ratio of 1:2:1. So you have one of this, and these two are the same as the genotypic ratio, and the phenotypic ratio will be given by 3:1. But if these alleles are separating independently of each other, we will get these four different combinations. So these four are for the father, these four are for the mother. Now you can make all of these combinations back again. So this individual is getting capital P from here and capital P from here, capital G from here and capital G from here. Similarly, take any other individual, let us take this individual. So this individual is getting small p from father, small p from mother, capital G from father, capital G from mother. So we can make all of these combinations, and in this case our genotype will be 1:2:2:4:2:2:1:1 if we add up all of these, and the phenotype will be given by all of these individuals will show the phenotype of capital P and capital G. This one, this one, this one, this one. So we will have four of these. So 1 plus 2 is 3, 3 plus 2 is 5, 5 plus 4 is 9. So we have 9 individuals that have purple flowers and green pods. Then the ones with white flowers and yellow pods is only 1. Now the ones with white flowers and green pod, now white flowers will be given by small p, so this is small p, and this is small p, and this is small p. But when we say green pod, it should have a capital G. So this has a capital G and this has a capital G. This does not have a capital G. So 1 plus 2 is 3. So there are 3 individuals that have white flowers and green pods, and similarly there are 3 individuals that are purple flowered and yellow pods. So yellow pod will mean that it has small g. So this one and this one. So there are 2 of these and both of these are having purple flowers because they have capital P. So 2 plus 1 is 3. So we have this ratio of 9:3:3:1. So this ratio is explained by the law of independent assortment. Now these are the example cases, but then we also see some variations to complete dominance in some cases. So we will have a greater look at these in the next lectures, but just to go with the definitions, codominance occurs when the contributions of both the alleles are visible in the phenotype, and a common example is the blood groups. So A, B and O. Now in this system we represent our alleles as IA, IB and IO. Now if there is an individual that gets an IA and IA from father and mother, we will call that this individual has a blood group of A. If you have IA and I0 or IO, so in this case this allele is expressing the proteins that are represented by A, and this allele is not representing any protein. So in the red blood cells we will get only one sort of protein, which will be A. Now similarly, if you have IA and IB, we will be getting this allele is representing proteins for A, this allele is representing proteins for B. So in this case we will get AB, and in the case of IO we will be getting an O phenotype. So in the case of codominance, what happens is that there is no recessive allele and there is no dominant allele, but depending on what combination you are getting, both of these will be able to express themselves. So in the case of codominance, both of these are equally dominant. In the case of incomplete dominance, what happens is that the phenotype of the heterozygous genotype is distinct from and often intermediate to the phenotypes of the homozygous genotypes. So what happens in this case is that suppose you have an allele that is capital R. Now capital R codes for a red color. So suppose it gives you a red pigment in the flower, and there is a small r that does not code for any color. So this does not give you any color. So there are no pigments here. So if you have a capital R, you have a double production of the red pigment. If you have a capital R and a small r, you have one time production of a red pigment, because small r is not coding for a red pigment, it is not coding for any color. And if you have small r, then there is no red pigment. Now in this case, this would look white in color because there is no pigment in the flower. This would appear say red in color, and this would appear a deep red in color. Or in circumstances, this would look like a red color, this would look like a pink color, and this would look like a white color. So in the case of incomplete dominance, these alleles are not completely dominant, they are somewhat dominant. So if you have a capital R and a small r, then you can see traces of R or the capital R in the form of red pigments, but then these red pigments are not enough to provide a complete dominant phenotype. So it will provide an intermediate phenotype which will look like pink or which will look like a light red color. So these were the definitions that we had to deal with. And why do we use conservation genetics in the field of conservation? So these are some things that we do in conservation genetics. We can go for a DNA cell line cryobanking in a frozen zoo. So this is something that we will look in our XC2 conservation module, what is a frozen zoo. Then chromosomal and karyotype analysis. So chromosomal and karyotype analysis will come in our chromosomal and genetic disorders. Then population genetics and management, which will come in the next lecture. Then we have DNA sequencing. So we saw that in the chromosomes we have all the information, and if we read out this information, we get a sequence that goes by the name of DNA sequence. So we can read out what is written in all of the chromosomes. Now DNA barcoding stands for selecting some regions of the chromosome so as to be able to identify the species. So for instance, if you look at a human being, if you look at me, you can very clearly make out that I belong to the species Homo sapiens. But suppose you only got a hair from my body. So whether this hair belongs to an individual of Homo sapiens or whether this hair belongs to some other species, how do you make out? So we make use of genetic analysis that goes by the name of DNA barcoding. So we have identified certain regions in the chromosome that are different between different species, and just by reading that small portion, or just by using some ways of identifying what that small portion says, will be able to tell what species it is. So just like a barcode is used in different products in a supermarket, so just by reading the barcode you can tell what the product is. Similarly, in the case of DNA barcoding, just by reading a small portion of the DNA, we will be able to tell what species it is. Or if we want to go into even greater detail, we can even say which population does this individual belong to, and so on. Next we go for genetic rescue and translocation. So genetic rescue is something that we saw before as well. It is a way of overcoming inbreeding, and we will look at it in more details in this lecture, reintroductions and outbreeding. Next is hybrid and parentage identification. So we can so suppose you have a situation in which you have wolves and you also have dogs living in the nearby. So you could have situations in which both of these species mate together and come out with an offspring. Now this offspring may or may not be a viable offspring, and this offspring may or may not be a fertile offspring. But then if you get some hair, you can make out whether this hair comes from one parent or whether this hair is coming from a hybrid. So such sorts of analysis and even parentage identification can also be done, and parentage identification becomes important in the case of small populations, in which case you want to prevent inbreeding as much as population. So this is something also that we will discuss in the population viability analysis. And then we also have phylogenetics and phylogeography. Now phylogenetics tells us how evolutionarily all these different species are arranged. So essentially when we say that all the life came out of say single living cells or cells like bacteria, how do we make out such kind of analysis? In the case of phylogeography, we want to answer the question what sorts of species and how is their interrelatedness when they are found in different geographical areas of the earth. So all of these are different topics that we deal with in conservation biology. So that's all for today. Thank you for your attention. Jai Hind.